Okay, so this is the Biology 251 Chapter 9 study points. So first we're going to begin with defining mixed and pure cultures and distinguish from each. So mixed culture is basically when there are microbes from different species that grow together on a petri plate, while pure culture is where a single species is isolated and all the bacteria that grows on a petri plate is going to be identical since they come from the same species. Define and explain what a colony is. So a colony is where there's millions of bacterial cells, but you have to know that it's from a same species. And colony morphology is basically where physical characteristics, like similar physical characteristics, like it's like opaque, it's white, it's round, and they just have the same characteristics because they're isolated from the same pure culture. And in pure cultures, they will have that similar morphology like I described, but in mixed cultures, you're gonna see different like morphology, like different colors. It's one's like translucent, one is opaque. Understand what binary fission is. So binary fission is basically where one cell divides into two identical daughter cells asexually. And basically, this happens either quickly or slowly based on species of bacteria from 20 minutes to 12 hours. Explain what exponential growth is. So, exponential growth is the basically the growth curve we see when we observe how long it takes a bacteria to grow, and it's gonna correlate with doubling time. So doubling time is basically when how long it takes a population to double and exponentially grow. Describe the steps of a growth curve. So a growth curve has 5 steps. The first one's going to be a lag phase. This is where the cells acclimate. So basically this is where we have little to no growth, but we see a little bit of growth. Okay, then exponential phase is where we see the large or sudden increase in growth. So there's going to be more growth than death in exponential phase. And then stationary phase is where after all that growth, the growth curve levels out and there's going to be an equal rate of death and growth. The death phase is where we see that decline from stationary phase. So the growth curve is declining down, and that's because we see more death of bacteria than growth. Phase of decline is just where it's that it eventually starts to level out towards the bottom of the graph. What kind of information can be derived from a growth curve? So growth curves are usually used to determine the growth rate of bacterial species. It's important to know what the growth rate is and to observe a growth curve because we can know how different environmental and nutritional factors affect the rate of growth. So for example, if like a bacteria was, if we were to test like its growth compared to a room temperature to a cold temperature, we can observe how fast that the bacteria grows with that growth curve. No one explained the different methods discussed in class for counting microbial cells. So direct plate counts. Direct plate counts is where you get a bacteria and you plate it onto an agar plate, and you do that over and over again to dilute it until you can count the colonies on the plate. Hemosidometer is a microscope tool that directly counts the number of microbial cells in a culture on a slide. A culture counter is an instrument that automatically counts individual cells via a detector. And a spectrophotometer, basically it measures the turbidity, so how dense a sample measures and how microbial cells are in a sample. When might the different counting methods be used? So direct plates are often used to diagnose UTI and determine colony forming units, CFUs. And a hemosidometer is when we're counting both living and dead cells, because we can see the living dead cells and the dead living cells and the dead cells on that microscope slide. And then a culture counter is when we don't have time to do all that and we need to count the amount of cells rapidly. Explain what a direct plate count is and how it is done. So a direct plate count is basically where we obtain that bacterial culture, like I said before. We plant it on agar plate enough times to dilute it enough to gather a countable number of colonies. And this is called a serial dilution. Know the different categories of grouping microbes according to oxygen requirements and utilization. This is a very important picture to know or be able to describe. So basically, an obligate aerobe is where a bacteria needs oxygen to grow. So if we look at this picture over here, it needs oxygen to grow. So oxygen will be towards the top of the tube is where it's exposed most to oxygen, right? So obligate aerobe would be at the top. All the bacterial growth would be at the top. And then an obligate anaerobe means it cannot grow in presence of oxygen. You can think of an like anti and aero oxygen, right? So antioxygen. So they can't grow in presence of oxygen. So oxygen will be less present towards the bottom of the tube, which is why obligate anaerobes grow towards the bottom of the tube and there's no growth at all at the top. Facultative anaerobe. So basically a facultative anaerobe, they grow in the presence of oxygen or without oxygen, but it prefers oxygen. So what we'll see is that there will be growth because it prefers oxygen, right? So more growth at the top, but there will be growth throughout the rest of the tube. A microaerophile is basically micro, so little, and an aero is oxygen, right? So they require little to no oxygen, but they still require oxygen, right? So there will be a little bit of space towards the top, but there will be more growth towards the middle of the tube and like none at the bottom. And then an aerotolerant anaerobe, it grows with or without oxygen, so it tolerates oxygen. Aero, oxygen tolerant, so oxygen tolerant. So basically, there will be growth throughout the entire tube. Explain how microbes tolerate ROS. So ROS is a reactive oxygen species. So what you need to know is that basically bacteria can like basically tolerate or have these certain enzymes. They have SOD and catalase, right? So if it has SOD and catalase, that means it likes oxygen. But if it cannot produce SRD or catalase, that means it does not like oxygen. And then if you do this, if you haven't done this in lab yet, you will soon, where basically you do a catalase test. And basically a catalase test is where you put the bacteria on a microscope slide and you see if it bubbles or if it doesn't bubble. If it has catalase, it will bubble because it can produce, it needs oxygen. That's how basically you confirm whether your species is obligate, facultative, and yeah. So basically, like I said before, if a microbe has SOD and catalase, it likes oxygen. So what microbe types like oxygen? We know that obligate aerobes like oxygen, so they will both have SOD and catalase. Facultative anaerobes, it can grow without the presence of oxygen, but it prefers oxygen, so it also have SOD and catalase. Obligate anaerobes don't like oxygen, right? So they won't have SOD and they won't have catalase. Aerotolerant, remember we say that it can tolerate the presence or the lack of presence of oxygen. So it'll have SOD but not catalase. And then microaerophiles need little oxygen. So it'll have a little bit of SOD and a little bit of catalase because we only need a little bit of oxygen. And then what is the catalase test used for? So like I said again, it's used to detect the presence of enzyme catalase in bacteria. That's why we do those tests. So if the enzyme breaks down hydrogen peroxide into water, yada, yada, yada. What you need to know is that if it's a positive, bubbling, that means catalase is present. But if there's no bubbling, that means catalase isn't present. So if we were to have bubbling, it would either be what? It would be an obligate aerobe, a facultative aerobe, or a microaerophile, because those have catalase. And what is the catalase test used for? Oh, sorry, I just went over that. We'll know about the Clostridium perfringens example. So a patient is diagnosed with gas gangrene, Clostridium perfringens in his foot. And basically, we're going to use hyperbaric, not hypobaric, hyperbaric oxygen therapy, which is high pressure oxygen chamber. And basically, that high pressure oxygen chamber kills C. perfringens because it's an obligate anaerobic bacteria. Obligate anaerobes hate oxygen, and if you're going to put it in a high pressure oxygen chamber, it's going to be instantly killed. Why? Because it hates oxygen. Be familiar with acidic and basic conditions and the pH scale. So 0 to 6.5 is acidic, 7 is neutral, 7 to 14 is considered basic. And how are microbes grouped according to acid-base tolerance in practical applications? So neutrophiles range from pH of 5 to 8. That's their neutral range. Acidophiles are going to have the pH below 5.5, and then alkaliphiles are going to have a pH above 8. And so that's going to be basic. Understand the details of the H. pylori example. So H. pylori gastritis is found in a patient's stomach and causes stomach ulcers. So H. pylori is not acidophile, it's actually a neutrophile, but it's not killed in acidic environment of the stomach. Why is that? It's because it has the ability to survive due to this ability to form urease. So urease basically creates like a bubble surrounding the bacteria that raises the pH to make a neutral microenvironment so that it doesn't die. Understand temperature requirements tolerance of microorganisms. So the optimum temperature depends on a species, but have a range of what temp it can grow within. So basically, it can grow from here to here, but if it goes outside of that range, it'll grow a lot slower. But it won't die, it'll just grow a lot slower. So a psychrophile, I think of psychos love the cold. So psychrophiles live in extremely cold environments. So think about like what is it called? Like the Arctic tundra. They can survive in those climates. And then psychotrophs. They prefer refrigerator temperature. So psychotroph, they will like grow within food stored in a refrigerator. That's why they're kind of important to know. Mesophiles, so they are going to be within our body temperature, and these are the pathogenic bacteria that we most often come across. Thermophiles, we think about like hot tub temperature, pretty hot but not like scalding hot. And then hyperthermophiles, extremely hot temperatures. Know the temperature category for classifying bacteria as practical applications. So, for example, like I said before, refrigeration is a form of preservation that slows down food spoilage. So, for example, like mesophilic bacteria, if they were put in a refrigeration tip, the growth would slow. But it will still spoil if taken out of the refrigerator. And freezing. Many microbes will be unable to grow besides psychrophiles, but this is not serialized food because as soon as you take it out of the freezer, it will start to grow again. Know about L. monocytogenes example. So Listeria monocytogenes is often grown due to improper food handling and sanitation. It's grown and ingested, and that's because Listeria monocytogenes is a psychotroph, right? So a psychotroph is where they like what's it called, the refrigeration tip, right? And this is why raw foods like vegetables and deli meats are the center of Listeria outbreaks. That's why we hear about Chipotle, iceberg lettuce Listeria outbreak, salami Listeria outbreaks. Understand solute salt concentration and food preservation. So halotolerant basically can tolerate higher concentrations of solute, while halophilic, so philic, remember that philic means love, require a higher concentration of solute in their environment. So think about like seawater. So an example of how food is preserved is that bacon is preserved with high salt concentration since bacteria undergoes plasmolysis. That's why we have like bacon bits that can stay in our pantry for months. And then another example are jams, honey, mustard, and ketchup. And note about the Staphylococcus aureus example. So Staphylococcus aureus is a common contaminant in salty foods, and that's because Staphylococcus aureus is a halotolerant bacteria. Therefore, it does not die in high salt environments. All right, and then understand and explain how bacteria are cultured in a laboratory. So we most know growth, nutritional environment requirements to successfully and optimally grow bacterial species, right? So we need to know the optimal temperature, and like moist environments, dry environments. If we're gonna have oxygen present, no oxygen present. That's why we use agar plates, agar slants, and broth tubes. And know the different kinds of growth media. This is a long list, but you need to know complex media is basically different mixture of different growth factors. Selective media is basically where we inhibit growth of a certain growth factor. And then differential is where we allow characteristics of bacteria to shine. So we basically find out whether or not it has this bacteria or not, or this bacteria has this characteristic or not. And then blood agar is basically a complex and differential media that basically determines whether bacteria can lyse red blood cells. And then McConkey agar plates. So McConkey agar plates are selective and differential. They select for gram-negative bacteria and whether it can ferment lactose. And these are the results. So, yeah. And that's it. Thank you.